Hello and thank you for watching the video of my presentation. Before starting, I wanted to mention that some part of the data I'm going to present here are already covered in our previous meeting. So I want to spend a lot of time on them. I will try to highlight some more recent finding and make a connection with the previous data. That being said, this presentation will cover a very brief introduction to sodium pump, its isoforms, its regulational, the mechanism that it regulates pump activity, and the function of the pump in a skeletal muscle. Then I will present the major findings from 4 manuscripts that I included in my dissertation, and if time allows, I will show some of the ongoing project and the future direction of this project. So sodium pump is a plasma membrane bound enzyme or pump that uses hydrolysis of 1 ATP to pump 3 sodium out, 2 potassium into the cell, and by its activity it establishes a steep ionic gradient across the cell membrane. So due to the activity of sodium potassium ATPase enables cells to concentrate ions. It also it controls a constant ion environment in the cell. The activity of pump regulates osmotic balance in the cell, which prevents cell burst. And interestingly, in membrane of the animal cell, pump activity enables the membrane flexibility, which eventually leads to the motility of organism. These are just part of a long list of cell-specific sodium pump function. The other function is that, for example, by establishing sodium pump gradient across membrane, pump establish membrane potential. And it also, in cells, in excitable cells like neuron and muscles, the sodium and potassium gradient preserve the excitability of cells. The other function of sodium pump is that the sodium potassium gradient across the membrane provides a driving force for the transport of glucose, calcium, proton, amino acid, neurotransmitter, vitamins, and other molecules through second transport mechanism. And it also provides the energy to support the electrical signaling by the means of action potentials. So it's not surprising to know that 50 to 70% of total energy used in gray matter of brain is just consumed by sodium pump activity, or 25% of total energy expenditure in the brain. So let's have a look at the sodium pump function and its subunit. Sodium pump comprise 1 alpha subunit, which is this one, 1 beta subunit, and in some tissue, a tissue-specific regulatory protein from the family of fixed protein, this one, this subunit. Sodium and potassium are transported by the catalytic alpha subunit. The beta subunit doesn't participate in ion transport, but it's required for assembly, maturation, and function of pump. It's also essential for translocation of the mature pump to the membrane. Fixed subunit is not required for pump function, but it plays a major role in regulation of pump activity, and importantly, it's a target of adrenergic stimulation. Several isoforms of each subunit exist. Alpha subunit exists in four isoform: alpha 1, alpha 2, 3, and 4. Beta subunit exists in 3 isoforms, and 7 isoforms of fixed subunit has been identified. Studies in expression system shows that all possible combination of alpha beta subunit has distinct kinetic properties like turnover rate, different sodium affinity, different potassium affinity, and also different ATP affinity. Since my project is about the physiological difference between alpha 1 and alpha 2 isoform, here I'm going to highlight some sequence similarities and differences between these two isoforms. So within a species, it turns out that the sequence similarity among alpha 1, alpha 2, and alpha 3 is between 80 to 90% within a species. Between a species, the sequence similarity among alpha subunits is at high 90%, which suggests a strong selection pressure for expression of different sodium pump isoform within an organism. As for tissue expression of sodium pump isoform, alpha isoform is the one that is expressed ubiquitously, and it's the only isoform that is expressed in kidney. Alpha 2 isoform is it predominantly expressed in skeletal muscle, heart, and brain. As for the subcellular distribution of alpha isoform, in cells with highly specialized structure and functions like neurons, heart, and skeletal muscle cell, isoforms of alpha 1 and alpha 2 are localized in different membrane domain. For example, in a skeletal muscle, alpha 1 only found in the surface membrane, but alpha 2 is localized predominantly in T-tubular system and some other specialized domains like neuromuscular junction. So alpha 1 and alpha 2 isoform have some evolutionary preserved differences in sequence, function, tissue expression, and subcellular distribution. But what is the tissue specific function of sodium pump in a skeletal muscle? First, a skeletal muscle is the largest body organ with 40% of the total body mass. It contains the largest pool of sodium pump in the body, and also the most of the total body potassium are stored within a skeletal muscle, up to 70%. As for the role of pump in muscle contraction, pump activity is required for action potential, and also during fight or flight response, the pump activity is upregulated and it delays fatigue and keep us running. But muscle has other metabolic roles and functions, like regulation of body plasma homeostasis is one of the functions of skeletal muscle. In this regard, pump activity of a skeletal muscle tightly regulate body plasma potassium, which prevents some dangerous low or high potassium level, which may lead to hyperkalemic periodic paralysis or even death. So understanding the regulation of sodium pump in a skeletal muscle is clinically very important because malfunction in activity of pump in skeletal muscle is attributed to several diseases like various types of muscular dystrophy, McArdle disease, and congestive heart failure. Skeletal muscle also a good model if you are interested in studying sodium pump activity, especially sodium pump isoforms, because skeletal muscle is the only tissue that expresses a large amount of alpha 2 beside alpha 1 in distinct domains of the membrane. It also contains the most dynamic pool of sodium pump. The activity of pump is regulated over a wide range in skeletal muscle, like from 5% of its total potential activity during rest up to 65%, more than 12-fold increase following electrical stimulation. The long-term goal of my study was to specify the role of different sodium pump isoforms in skeletal muscle function. So as a first step, I developed a method to quantify sodium pump activity in intact myofiber isolated from skeletal muscle, which is a primary cell. So before introducing the method, let's see how pump activity is typically measured in pump briefly. So sodium pump hydrolyze ATP to pump out sodium and pump in potassium and to polarize the membrane. So it can be considered an enzyme or pump or a charge transporter. To quantify the activity of pump, we can measure the amount of ATP that is consumed, which gives us the enzymatic activity of the pump. We can measure the amount of sodium decline within the cell or sodium increase introduced into the cell, or the magnitude of the negative charge that is generated in the membrane. But each of these methods has come with some interfering factors. For example, in the case of a skeletal muscle, if we are going to measure the amount of ATP that is consumed, there are other motor proteins like myosin that use a lot of ATP. So to get rid of that interfering factors, we homogenate muscle and purify membrane. Or in case of if we are going to measure the charge generated in the membrane, there are other charge carriers like sodium and potassium channels. So we have to block all sodium and potassium channel to be able to measure the specific activity of pump. If we are going to measure the amount of potassium that is introduced to the cell by the pump, there are high potassium background in the cell that reduce signal to noise ratio. So we have to we typically use rubidium, which is as a surrogate for potassium or as a tracer for potassium transport. For some experimental reasons that are beyond the scope of this presentation, the rubidium uptake is considered the gold standard method for quantifying the sodium pump activity. But this method also has its own limitation that we described in this paper. And we introduced a new model for quantifying the activity of sodium pump in a skeletal muscle. In this method, I describe how to isolate myofibers from EDL muscle and how to measure rubidium and other ion signals by ICP-MS method. By the way, this is the mass spectrometry based method. ICP-MS stands for inductively coupled plasma mass spectrometry. And we also show that how to use sulfur and phosphorus signal as a normalizing factor for for heterogeneous muscle cells in terms of cell volume and cell count. So we showed that it's valid to use these signals as a normalizing factor. What's the advantage of this method? The traditional uptake method, in traditional uptake method, you have one EDL with a whole muscle, different type of muscle, but each of the muscle provide a single data point. New method based on myofiber, in new method based on myofibers that are introduced, you have 100 myofibers from one mouse, which eventually reduce the number of animals that required for each experiment. And it also provides a more homogeneous muscle group, which increase the statistical power of the test. So it's kind of mid-throughput method that is very useful when testing the dose dependency of drug or stimuli effect on pump activity. Moreover, ICP mass spectrometry allowed us to use naturally abundant rubidium instead of radioactive isotopes of rubidium, which is safer and cheaper and it's easily scalable. The next project that I'm going to discuss here is about the acute regulation of sodium pump isoforms by potassium concentration. But before that, let's review some general mechanisms of pump regulation. The pump is regulated at 3 levels. First, at the level of intrinsic activity of pump, or how fast or slow the pump turnover rate is, or by abundance of pumps in plasma membrane, or by the third mechanism, or by a total content of pump in a skeletal muscle. Intrinsic activity of pump depends on the concentration of enzyme substrate in this case sodium, potassium, and ATP. It also depends on protein-protein interaction, like interaction of pump with phospholamine protein. More on that later in this presentation. And also depends on the post-translational modification of pump, like phosphorylation or glutathione. The abundance of pump in plasma membrane is controlled by translocation of isoforms or a pump between subungular compartment like ER or surface membrane or T-tubule system, as I mentioned before. And the main mechanism to control total pump content in a skeletal muscle is by transcriptional regulation of pump subunit expression, and to a lesser extent by regulation of translation and protein stability and epigenetic mechanism. In our 2015 JGP paper, we studied how potassium concentration regulates the intrinsic activity of pump isoform, and we demonstrated that the potassium sensitivity of alpha-2 isoform is much lower than what previously suggested. So in this paper, we measured the activity of pump with two methods. First, by measuring potassium sensitive current in voltage clamped myofibers that are extracted, that are isolated from flexor digitorum brevis muscle from mouse foot, or FDB muscle myofibers. And the second method was to measure K-ATPase activity in mixed hind limb muscle from mouse. The mouse model that we use in this study was Y-type and skeletal muscle alpha-2 knockout, which is a targeted knockout of alpha-2 in skeletal muscle. And in this model, alpha-1 is the only active isoform in skeletal muscle. One major finding from this study was that in terms of potassium affinity of sodium pump, K-half of alpha-1 is significantly smaller than K-half of alpha-2, we can say alpha-1 is more potassium sensitive than alpha-2. I will discuss the physiological implication of this finding later in this presentation. In another related study that we published in 2018, we aim to obtain a more detailed information on the dynamic effect of the plasma potassium on sodium pump isoforms. So in this research, we use different animal models and we enhanced our technique, and we further validated the specificity and sensitivity of our myofiber or ICP-MS based method. To measure potassium sensitivity of pump isoform, here we use myofiber technique that I developed. Myofibers were obtained from extensor digitorum longus muscle, this part of mouse foot, from 3 mouse model: wild type, skeletal muscle alpha-2 knockout, and swap mouse. So what is the swap? One significant difference between alpha-1 and alpha-2 isoform is their sensitivity to wobbin or digoxin, which is a specific pump inhibitor. Alpha-2 is wobbin sensitive in wild-type rodents, and alpha-1 is wobbin resistant. So with treatment with low concentration of wobbin, you get alpha-1 as the only active isoform of the cell in wild-type rodents. It's very useful property when you are studying isoforms, but it allows studying alpha-1 specifically, not alpha-2, because eventually the low concentration of wobbin inhibits all alpha-2. This issue was resolved by introduction of swap mouse, but by Dr. Lingrel and his team. So in this animal model, alpha-2 is the wobbin resistant and alpha-1 is the wobbin sensitive. So with low concentration of wobbin, you get alpha-2 as the only active isoform, and high concentration of wobbin eventually blocks all isoform in all model, which is a useful property to separate a specific versus non-specific activity of pump. So we determined the specific potassium kinetics of sodium pump isoform in skeletal muscle by using myofibers from wild-type alpha-2 knockout and swap mouse in combination with different concentration of wobbin. For example, here we have we increase potassium and we measure rubidium activity in wild-type animal, which gives us the potassium affinity of alpha-1 and alpha-2. Wild type treated with low concentration of wobbin, alpha-1 potassium affinity. Swap mouse plus low concentration of wobbin gives us alpha-2 potassium affinity, and skeletal muscle knockout contains only alpha-1 potassium affinity. Gives us potassium affinity of alpha-1 only. And the result from all 3 animal models are consistent and suggest that the potassium affinity for alpha-1 is significantly higher than potassium affinity of alpha-2. Here we have alpha-1, alpha-2, again alpha-1 and alpha-2. In terms of the maximum activity of pump, the maximum activity of alpha-1 is significantly lower than alpha 2, probably due to the lower membrane abundance of alpha 1 in skeletal muscle. So what's the physiological implications of the difference in potassium kinetics? Let's go back to the localization of the sodium pump isoforms in different membrane domains. As I mentioned, alpha 1 is expressed in surface membrane, alpha 2 in tubular system and neuromuscular junction. So one important thing is that muscle contraction requires a continuous train of action potential. Each single action potential introduces a high amount of potassium to the interstitial space here and also here. So at the surface membrane, this extra potassium immediately diffuses to plasma space. But in T system, the extra potassium cannot diffuse and it accumulates. So why T-tibial system are diffusion limited system? First, because of these narrow T-tubules, and second, because of the tortuosity, which is a feature of disordered networks like T system, and it's unlike ordered system. Unlike ordered system, these tortuous system are diffusion limited. So with K half of less than 1 millimole, alpha 1 isoform is already saturated at the resting level of extracellular potassium, which is 4 millimole. There is no more room for upregulation. As potassium is accumulated at the lumen of two-tubular system, it stimulates more alpha 2. So we can say that accumulation of potassium in T system regulates alpha 2 through stimulation, but doesn't have any effect on alpha 2, which is already saturated. This is consistent with the membrane localization of alpha 1 and alpha 2 and kinetics property of two isoforms. So my next project was about regulation of sodium pump by adrenergic stimulation. During fight or flight response, all the mechanism that leads to fast and continuous muscle contraction are self-limiting processes. So with a fixed sodium pump activity at the similar to resting level, it may lead to impaired muscle contraction and early fatigue. So we need maximum activity of pump to be able to run, which is the role of adrenergic stimulation. What is adrenergic stimulation? A short-term reaction of sympathetic nervous system in response to external stressor by sudden rise of catecholamines, or including epinephrine and norepinephrine. Both of these neurotransmitters and hormones are shown to increase or decrease pump activity. In skeletal muscle specifically, the fatigue resistance effect of adrenergic hormones, epinephrine and norepinephrine, are suggested that their action is through activation of sodium pump turnover rate. But the kinetics of this action and the isoform target of adrenergic effect is not well known. In this manuscript, I did some contractility experiment and also I measured pump activity to determine the kinetics and sodium isoform target of adrenergic effect. Here what I did is that I electrically simulated a whole muscle with 60 Hz frequency for 60 seconds, and I recorded the time course of force decline. This is force and this is time. This is force and time. So three contractile parameters was calculated based on this recording: maximum force, the time to half maximum force, and the total area under curve, which represent the total force generated over stimulation time. The central question in this experiment was that what is the effect of adrenergic stimulation on muscle contractility? And what is the effect of high extracellular potassium on muscle contractility? Is there any interaction between these two factors, adrenergic stimulation and high potassium? And what is the role of sodium pump, alpha 1 or alpha 2, in response to these two types of stimulation? To separate the contribution of alpha 1 and alpha 2 activity in muscle contraction, I conducted this experiment in 12 different conditions. So we used muscle from wild type and swap animal by varying wobin concentration. For the sake of this presentation, I used these blue bars to show when we had normal potassium, extracellular potassium, and we had high extracellular potassium. These red lines represent salbutamol treatment. Salbutamol is a synthetic adrenergic agonist, beta-adrenergic agonist or adrenergic stimulator. The gray line represent the baseline or no adrenergic stimulation, and this shows which isoform was active during the experiment. So in wild type without wobin, which we have alpha 1 and alpha 2 as active isoform, with normal concentration of extracellular potassium, the adrenergic stimulation has almost no noticeable effect. Compare red versus gray here. But with high extracellular potassium, this experiment, the high extracellular potassium itself impairs contractility. Compare red versus gray here. And salbutamol treatment rescues contractility almost 100%. It recovers, it's back to normal. When we have wild type plus micromolar wobin or alpha 1 active only, with the normal concentration of extracellular potassium, the salbutamol treatment slightly enhanced contractile profile, but it's not the big boost. When we have high extracellular potassium and alpha 1 active only, the high potassium impairs contractility dramatically. Just compare gray here with gray here. And the effect of adrenergic stimulation here is more pronounced, like 2 to 3-fold increase in the total force generated after adrenergic stimulation. When we have swap plus wobin, which gives us alpha 2 activity, again during normal potassium concentration, the adrenergic stimulation slightly enhanced contractility, but when we have high potassium and alpha 2 active only, the high extracellular potassium has no noticeable effect, and also adrenergic stimulation has very slight and non-significant improvement. Here what we have is 3 different contractile parameters: peak generated force, area under the curve, and time to 50% of the maximum activity. For example, here I indicated peak generated force. In 12 conditions, we have normal and high potassium we have without or with adrenergic stimulation, alpha 1 and alpha 2, alpha 1 only and alpha 2 only. Similarly, for time to 50% of the maximum force generated, and the area under force time curve. I've listed some noticeable findings from the EDL contractility experiment here, but I just wanted to mention that overall our conclusion is that alpha 1 and not alpha 2 is the primary target of adrenergic stimulation. There are many confounding factors that may involve in the effect of high potassium and adrenergic stimulation on muscle contractility that I measured, because muscle contraction is a multifactorial, very complex process. For example, potassium-induced membrane depolarization may or definitely affect contractility of muscle. Or another confounding factor is that high potassium selectively increase or stimulate alpha 2 isoform, as I mentioned before. So to eliminate these confounding factors, I started measuring sodium pump activity in 18 different experimental conditions directly. This is not contraction. This is rubidium uptake or sodium pump activity directly measured in myofibers in 18 different experimental conditions. So what we have here is the rubidium uptake by alpha 1, alpha 2, alpha 1 and alpha 2, or non-specific ion transporters, non-pump transporters, in high versus low potassium, with and without adrenergic stimulation. So alpha 1 activity, let's review one by one. Alpha 1 activity is upregulated by sodium, and regardless of potassium content. You see that potassium doesn't has any effect on alpha 1 activity. In alpha 2, and conversely, in case of alpha 2 isoform, salbutamol do not affect alpha 2 activity, but high extracellular potassium do increase rubidium uptake. Then we have alpha 1 and alpha 2. These contradictory effects probably cancel each other, and that's why we don't see any significant change. Another interesting effect of adrenergic stimulation studied in this project was that it changed the temporal dynamic of pump activity. Here we have time versus pump activity after adrenergic stimulation. So in panel B, you see that salbutamol affects the time-dependent activity of alpha 1 and alpha 2 differently. This is alpha 1 and this is alpha 2, and you see that the time-dependent activity of them are changing, the dynamics of them are changing following adrenergic stimulation. But when I measured ATPase activity in purified membrane, I got somehow contradictory result. Here, there is no considerable difference between alpha 2 versus alpha 1 in terms of response to adrenergic stimulation. Both of them follow almost similar pattern. There are many explanations for this discrepancy, which we discussed in the manuscript, but the one that I want to mention here is related to the localization. localization of alpha 1 and alpha 2 in the membrane. So skeletal muscle cells are very large cells, and they are highly compartmentalized. And some cellular functions in a skeletal muscle depends on the spatial access of one pool of protein to another pool or another domain of the membrane. It's been suggested that in myofibers, cyclic AMP, which is the core component of adrenergic stimulation, the cyclic AMP production and pKa activity are localized to different subcellular compartments. Therefore, the different adrenergic sensitivity of alpha 1 and alpha 2 might be due to the asymmetric localization of alpha 1 and alpha 2, which provide a spatial with different this alpha 1 the different localization means different spatial access to signaling complexes and receptor proteins. So most of the data I presented today suggests the different roles of alpha 1 and alpha 2 isoform of sodium pump. But the central question is why we might need different isoforms, or why several isoforms of the same ion transporters are well preserved during evolution. In terms of sodium pump isoform, the general consensus in the field is that alpha 1 isoform play the main housekeeping role, and alpha 2 and alpha 3 are only activated during a stress condition, or they are involved in the fine-tuning and regulation of cellular function. This is also a common pattern that is observed for many other protein isoforms. You knock out the one isoform, you put organism under the normal condition, and mostly there is no effect or very mild effect. You put organism under a stress condition, and the true function of that isoform appears because the organism cannot tolerate this specific stress condition. In this view, isoforms are seen as a reserved capacity. But some of the results that I presented here, such as potassium affinity, different potassium affinity of alpha isoform, different role of sodium pump isoforms in contractility and high potassium response, and also the isoform specificity of the adrenergic stimulation among alpha 1 and alpha 2 isoform. These results may imply that some isoforms, like sodium pump alpha 2 isoform, are evolved for a distinct function, for a different function, and are not just a reserved capacity for a stress condition. These are some clinical significance of this study that I don't think I have time to review it, and that's all. Thank you very much for watching this presentation.